Good morning, everyone in YouTube land. And let's go ahead and get the thumbnail over with. Okay. I honestly don't know. But, hey, we're finishing up. So, let's see. We're going to discuss today a little bit about metastasis and the role of oncogenes in cancer and tie in actually a lot of this stuff we've been covering. And then finish up chapter 20 it is. I will start posting lecture notes, sort of things like I've done before. And I'm thinking probably on Sunday I'm going to record a general review sort of discussion about just what the exam's going to be. But in general, it's going to be the same as the other two. Format will be pretty much the same in that we will open it at the final exam time, which I think is at 9, and close it at 11. And hopefully for everyone that'll work. If not, give me a holler. There was some discussion, yes, that chemistry was...
Any possibility we could open at 8?
Sure.
Great.
All right. Open at 8, go to 11, even better. Great. Yep. All right, so I'll, as I say, on Sunday I'll go ahead and do a little song and dance lecture. I may even bring Twinkle. And see where that goes, okay? So, we've been talking about the extracellular matrix and cell connections, the way cells interact with each other. And this sort of next step is what happens when cells are misbehaving. You have the paper on model systems for metastasis, which is sort of the advanced stage of cancer development, and that we're about to get into this here, but cancer cells, when they begin to divide very rapidly, they'll begin to pick up mutations that will allow them to actually uncouple from cell-cell connections and actually invade through the extracellular matrix and spread, and this is a very serious concern. But to the point, so we've been covering the different kinds of junctions, and we've got tight junctions that are essentially sealing up the epithelial sheet. There is not a whole lot of cell adhesion or structural elements. The cell adhesion point isn't being transferred into the cytoplasm. And then we have adherence junctions, which are attached to actin. And because, of course, actin has a motor protein, myosin, non-muscle myosin in all cells, this can serve as a way of changing cell shape. And the ability of an epithelial sheet to curve and change shape is transmitted through adherence junctions. A desmosome and its associated hemidesmosome are cell-cell or cell-extracellular matrix interactions, and with them we have intermediate filaments inside. And so this is providing a tremendous amount of cytoplasmic structure because the intermediate filaments are quite strong. While they don't have motor proteins, they are providing a pretty significant amount of structure. And last but not least, we have the gap junction, which is not really a cell adhesion type thing. It is, in fact, generating a cytoplasmic connection, where we actually have the connexon autocatalytically embeds in the membrane and generates a tube that allows for a cytoplasmic connection. And we can actually determine this from a couple of experiments. One experiment that's done is you can inject dye in one cell of an epithelial sheet, and if there's gap junctions present, you can see that dye diffuse across. Another experiment that's been done is looking at the conductivity of an epithelial sheet. In that individual cells, if they don't have gap junctions, they got a pretty good insulating layer of plasma membrane. And so if you were to take an electrode, very teeny, and stick it in this cell, and take another electrode and stick it in that cell, you can determine the electrical conductivity across that cluster of cells. If there are no gap junctions, that electrical conductivity is going to be very low, the resistance will be very high. But you see when gap junctions form, the electrical conductivity is very low, indicating that there is in fact a cytoplasmic ionic current that can pass from one cell to the next. So just to summarize it up, people then immediately began assaying what was the size of molecule that could pass between gap junctions. And so they're fairly small, such that metabolites can move back and forth. Ions can move back and forth. Large proteins, nucleic acids, no. But cyclic AMP can move back and forth, and we know the significance of that, and that cyclic AMP is a potent activator of a whole range of different kinase systems. And so you can have cyclic AMP being generated by adenylyl cyclase in this cell, and then activating things like protein kinase A way over here. So it's just another way of cell organization, and just one final point. All right, so on then to the idea of cancer and oncogenesis. So tumor production in cancer cells are the result of some kind of mutation. And a couple of bits of terminology here. We have when cells begin to divide in an abnormal way, and they begin to behave in an abnormal way as they're moving towards the development of a tumor. This is referred to as the transformed state. So a cell that is considered a cancer cell has been transformed. And the transformed state has a couple of very specific hallmarks to it. And when cancer diagnoses are being made, there will be a tissue biopsy, and a trained cytologist will look at the cells. And some of the things that they're looking for is an irregular pattern to the epithelium, or the overaccumulation of membrane. There are a couple of different kinds of things. You may be familiar with the idea of what's called a pap smear, which is a collection of cells, and a pap smear does not have to be actually uterine wall cells. It is a stain type. And it's actually the Papanicolo stain is really over 100 years old. It's a fairly old style way of staining cells, and it takes a trained cytologist to now look at those cells and determine whether they are in a normal epithelial sheet or if they're beginning to behave strangely, which is the indication of a potential tumor. So there are all kinds of now modern genetic methods and labeling methods that are also done, but the just observation of cells is a good place to start. So the transformed state has two key primary indicators. One is what's referred to as a loss of contact inhibition. And this is kind of why this topic is brought in right now. Contact inhibition is a phenomenon that animal cells will exhibit, in which if you were to take a collection of epithelial cells from the inside of your cheek, you could put them into culture, and they'll probably divide for a week or so because they're not transformed. And you add antibiotics to the cell media so you're not getting any bacteria and stuff growing. The cells will divide, and they will come to a spot where they are confluent. One of the things you can do is take a little rubber spatula and take a scrape down the middle of the tissue culture plate so you have a gap. The cells around that gap will begin to divide again, but they will touch, and when they have made connection, when they contact another cell, they stop dividing. And this happens because there are the cell signaling links that are associated with the different kinds of connections. And so we look at integrin as one example, that when integrin interacts with fibronectin, that's an indication that you have landed on a surface, and there are integrin-associated kinases that say, hey, we're on a cell surface. The same thing happens with some of these different kinds of cell adhesion molecules. They have kinases associated with them. So when the cells divide to a point where they touch, that cell knows, I've made contact, let's stop dividing. A loss of contact inhibition on the tissue culture plate will result in something that starts to look like this, where you have the cells have connected, they've contacted, and then they start to pile up on top of each other. So this loss of contact inhibition is a sign that there are systems in the cell that are no longer either providing the signal or no longer being paid attention to. And every time I have a little cut, a little boo-boo, I always say to myself, okay, the cells are going to fill in, and when they touch, please stop dividing. And if you ask them nicely, they will. So that's loss of contact inhibition. That is one of the hallmarks of a transformed cell. The other hallmark of a transformed cell is, this is an old-fashioned term that is used, but I think it's useful because this is what the standard transformed state is defined as. It's something that's called, cells have the ability to form foci in soft agar. Now this is, I don't think, a standard testing process, but it again underscores the fact that the cells are no longer paying attention to their environment, in that this term foci indicates a cluster of cells. Soft agar is, as the name implies, it is a very soupy agar, not like an agar plate. Boing, it is actually semi-liquid. And a non-transformed cell will not grow unless it touches a surface. So tissue culture plates that you want to culture primary cells are coated with gelatin, a component of the extracellular matrix. You can get them coated with collagen. They are coated such that when the cell lands, integrin and other kinds of molecules will bind and signal the kinase that says, we've touched down. In a transformed cell, they don't require that connection, and they actually can divide a little bit like a bacteria cell. The bacteria do not require a matrix to land on, but a non-transformed cell will. So, just as an example, when you're working with animal cell tissue culture, non-transformed cells, and it depends on the different kinds of cell cultures, but they require some kind of matrix to grow on. And transformed cells actually will grow in a shaker flask, whoosh, 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 in liquid culture. So when you're talking about tissue culture cells, these are very often cells that are somewhat transformed. If you were to take your epithelial cells from your cheek and put them into culture, they're only going to grow for a few days, a week or so, and then they stop and they're kaput. When you're working with animal tissue culture cells, and those of you that know Dr. Jen, she has a set of tissue culture cells that she works with, and you may have heard of what are known as HeLa cells, which are named after the woman that they were isolated from an ovarian tumor, Henrietta Lacks. Actually, a really interesting story. Those are cells that have been transformed, and they are now immortalized. They grow forever. There are no immortalized cells like that in our bodies. It's very difficult to find a cell that would continually grow under its normal genetic constraints. You start to pick up a few mutations, and they will begin to grow. Tissue culture cells are grown in the lab, animal tissue culture cells. They have all undergone some level of genetic modification, mutation, and such that they're able to grow in continuous cell culture. Some of them have developed sufficient mutations that they really are cancer cells. Others have developed just enough mutations to allow them to grow as an immortalized cell line. There's all kinds of different cell lines around. But normal, standard cells won't grow in that kind of condition. All right, so let's think about and look at what's happening that causes cells to become transformed. And not surprisingly, it involves cell signaling. So one of the big experiments that was carried out back in the 70s by two guys that ended up winning the Nobel Prize, Bishop and Varmus. I think it's Stephen Bishop and Harold Varmus. But just like Doberstein, I might be wrong. Anyway, Bishop and Varmus were one of the first guys to begin looking at why cells are becoming transformed. And one of the tools they used was something that's referred to as a transforming virus. We haven't talked a whole bunch about virology. We do that in molecular genetics, but Viruses, of course, are able to get into a cell, and certain viruses can integrate their genomes into the host genome as part of their life cycle. And it turns out that if you go digging around in the animal kingdom, there are a number of animals that have a virus that can cause cancer, a transforming virus. And it's interesting and fortunate that there are not a lot of viruses that humans can contract that actually cause cancer. Human papillomavirus is close to it in that there's not a guarantee of generating different kinds of ovarian and uterine cancers, but it is strongly associated with that, which is why there is now the vaccine called Gardasil, which is a vaccine against human papillomavirus. HPV ends up being associated with a lot of different kinds of uterine and ovarian tumors and various tumors in the genital urinary tract in both boys and girls. But in some animals, how many of you have kitty cats? How many of them are vaccinated against feline leukemia virus? Your cat has got to be vaccinated against feline leukemia virus. Just making sure, as a public service announcement, feline leukemia virus is a virus that is transmitted much in the same way that a cold virus is transmitted. Yet, when a cat gets the feline leukemia virus, they don't get the sniffles and clear it, they get leukemia. Which, let's just think about this, is really fortunate that there are no major transforming viruses within the human population. That would be a very scary thing if instead of COVID generating a cold, it generated leukemia. Not a good thing to think about. So that's just an example. There are transforming viruses in monkeys. There's lots of transforming viruses in chickens and in mice. And these are viruses that have been studied, and they have the ability to generate specific tumors. Things like the avian myeloblastosis virus causes muscle tumors. There is the simian viruses that can cause tumors in monkeys and things. So transforming viruses are known. Bishop and Varmus set out to figure out what was the transforming factor in this case that was actually causing cells to go into the transformed state. And the experiment, we're not going to get into too much, but what they did was they took a chicken virus and had chicken tissue culture cells. They kind of busted apart the DNA that was in the virus, infected the cells, or actually transfected the cells with this DNA, and looked to see which cells began to take off. And they could identify a fragment of the virus that was, in fact, the transforming factor in this case. And this was a gene that they referred to as an oncogene. So in Bishop and Varmus's initial experiment, they identified a gene that was in a transforming virus that if the host picked that up, that would cause transformation. So it's referred to as an oncogene. A little bit further study showed that what they called the oncogene, or their terminology became this, is they referred to it as the VONC, or let's do it this way, the viral oncogene. It had a closely related, but not identical, homologue in the host genome that they had to now call the cellular oncogene, C-onc. And so what they've discovered is the fact that oncogenes are us, that we have oncogenes, but we have them in the wild-type normal form such that they are under appropriate control. And what they discovered was you can add a viral oncogene to a cell, and that is what generates the transformed state. So this actually opens the door to study of oncogenes. And the kind of gene that was involved in all these different viruses begins to be identified. And in every case, it turns out there is a cellular homologue. And the mutations can be quite wide-ranging. When you compare the viral oncogene to the cellular oncogene, a couple of things you see are different kinds of mutations. There can be essentially, because we're talking about the DNA, a reading frame fusion. You might have some kind of deletion. You could even have things that are fairly small, like point mutations. There are a range of different kinds of phenomena that are observed that turn a cell signaling protein into a oncoprotein, a cell signaling gene, a gene that codes for a part of the cell signaling system, into oncogenes. So, just as an example, one of the things we have seen is this. If you recall, when we were talking about the old RTK class of signal transduction systems, the receptor tyrosine kinases. If you recall, we diagrammed this like this. They don't have a G protein associated with them. The receptor is generally a dimer. The hormone is also often a dimer. When the hormone binds the receptor as a kinase phosphorylates its neighboring partner. So it's a cross-phosphorylation. Here, how are you? Have a phosphate. Oh, hi, how are you? Have a phosphate. So there are multiple phosphates attached. And I don't insist anymore, but you know, whenever we said phosphate, what were you supposed to do?
Big negative.
Thank you. For to jump up and say negative charge, but really that's perhaps getting a bit excessive. So these phosphates and their massive negative charge are sitting out here, and they recruit other signaling molecules that themselves are not necessarily kinases, but they are going to be activating other kinases. And one of the very early discovered proteins that is recruited in the receptor tyrosine kinase cascade is a protein that we call RAS. And you saw this perhaps in the diagram that we were looking at. RAS is an oncogene that is the Rouse avian sarcoma virus. And that's how it gets its name. So there is the RAS oncogene, there's the FES oncogene, there's all kinds of different oncogenes. RAS is a protein that is involved in transmitting the signal from the phosphorylated receptor tyrosine kinase, and in turn, it's going to activate a variety of other downstream signaling genes, not so much amplification, but eventually we are going to have some kind of kinase activated. Another system that was like this was the MAP kinases. These are the mitogen activated protein kinases. And that's the one where we have the MAP kinase and the MAP kinase kinase and the MAP kinase kinase kinase.
Is the cross-phosphorylation going on up top above, or is it down below with all the phosphates?
Oh, the cross-phosphorylation is occurring on the cytoplasmic side.
Okay.
Yeah. The receptor, the external side, simply binds the hormone. It doesn't involve phosphorylation. The kinase activity is inside.
Cool.
Yeah. All right. So we have actually, and the name then again implies this, mitogen activated protein kinase. Mitogens are compounds that you add to cells that cause them to start dividing. You don't want to stick your fingers into things that are known mitogens. One of the things I've been kind of digging around with as my wife has been doing her herbalism study is a plant product that is generated by the euphorbs. And these are things like, what's a good euphorb? My mind has just drifted away. It produces something called 4-ball esters. And 4-ball esters, they're a plant secondary product. Okay. 4-ball esters will activate mitogen-activated protein kinases. 4-ball esters act as a trigger for cell division, and they do it because they're turning on a class of kinases that, because they triggered cell division, were referred to as mitogen-activated protein kinases. Compounds that will stimulate cell division abnormally are referred to as mitogens, mitosis. All right. Forbs. Anyway, there you go. The Euphorbiaceae. All right, so those are then potential carcinogens. Ralph-Sabian sarcoma virus, its name is now enshrined in that all of us have the RAS gene. In fact, in just about every cell in your body, you've got the RAS gene. And in its normal state, it is very tightly controlled. It's a good doggy. It maintains itself on its leash. When the doorbell rings, it barks for just a few moments, and then it stops. You get a little bone. It's all good. A mutant RAS is barking all the time. Doesn't require the doorbell to ring. It can't be silenced. And that's how you end up with an activated oncogene. So the different kinds of mutations then tend to be mutations that are involved in cell signaling proteins that cannot be turned off. And that is a unfortunate thing. So we've got and this can be actually in a whole range of cell signaling systems. RTKs are a good example, but there are oncogenes associated with all kinds of cell signaling systems. So you end up with one of these different kinds of kinases that are very potent activators, and it is critical that you can turn them on with precision, and just as critical that you turn them off with precision. If they're left on too long, they begin to cause difficulties in the cell metabolism. So the mutations then, we end up having, in some cases, a kinase domain portion of a gene can be transposed, can be moved around somewhere where it's no longer under the appropriate control. We can have a point mutation. A classic one would be you have a neutral amino acid. We'll pick up a mutation that will make it so it is now no longer neutral, but carrying a negative charge, like it goes from being a serine to an aspartic acid. And it turns out the genetic code makes it so that's kind of hard, but that can happen. And you have a neutral amino acid that is going to be the site of a phosphorylation with a big negative charge. What if it gets turned into an aspartic acid? That aspartic acid's got a negative charge. Now, it's not as big as the phosphate negative charge, which has got a negative charge like that, so you can see graphically, of course, but it is sufficient that it activates a little bit, and we now have unregulated things starting to happen. Now, what happens then is as a cell picks up a mutation, it begins to divide more rapidly, and it begins to provide a target for more mutations. And as people have begun to study more and more about oncogenesis, and particularly we start linking in genetically inherited cancers, like familial inherited breast cancer, we begin to see that there are two classes of cancer-causing proteins, cancer-causing genes. We've got things associated with cell signaling, very often, that are referred to as the oncogenes. They've got to pick up specific mutations that activate them. So the standard mutation is probably not going to do anything particularly useful for a gene. I mean, it's a little bit like saying, I'm going to open up the trunk of the hood of my car, and I'm going to pull a random wire. It may not do anything, but it probably is going to do something like not let you start your engine. It probably is not going to do something good for you. Hey, the air conditioner works better. I get better radio reception. No. Most of the time, a random mutation is going to probably cause a stop codon or a splice junction mishap, such that the protein is no longer functional. That's good. In certain mutations, in certain genes, it's not that it makes it non-functional. It actually makes it now so it can't be controlled. It's a gain of function mutation. They're fairly rare, but they do happen. What this means is oncogenes, typically, if they're getting mutated, they are not going to work. It would be a recessive mutation. But there are specific kinds of mutations in these genes that can make them now act as dominant mutations. Meaning that, try to throw back to genetics here, meaning that you have one wild type copy, a normal copy, and then the other copy on the homologous chromosome has picked up a mutation that makes it so it's no longer being, it no longer requires phosphorylation, or it can't be dephosphorylated, or it's on too much. This is a dominant mutation because the normal copy isn't able to make up for the effect of the mutation. And so you now have one wild type copy and one dominant mutation copy. And so even though you still have the functioning copy, the dominant mutation is turning things on. It'd be like, you have two doorbells and you can typically ring one or the other, but one doorbell gets a little bit funky, you hit the button, that is the hormone touches the receptor, bing, and instead of shutting off, the doorbell's on all the time, makes the dog crazy. This one's still, ding dong, still working. Didn't make any difference. It is a dominant mutation. The other class of genes that are responsible for a full-blown transformed state were a class of genes that took a little bit longer to discover. And these are referred to as tumor suppressor genes. Tumor suppressors typically are DNA binding proteins. They are proteins that are responsible for modulating expression of a whole range of genes. Just a couple of tumor suppressors to throw out here. One, perhaps, one of the very first to be discovered is a protein that doesn't have a gene and a protein that doesn't have a very evocative name. It's called P53. P53 was one of the very first tumor suppressors. It turns out that there are a bunch of tumor suppressor genes. You may be familiar with the tumor suppressor gene that is associated with familial inherited breast cancer. It is the breast cancer associated gene known as BRCA1. BRCA1 is another tumor suppressor. There is a childhood kidney disease known as a Wilms tumor. And the Wilms tumor gene is, in fact, a tumor suppressor known as WT1. All right, so tumor suppressors, these have to be mutated as well in order to carry a cell into a full-blown transformed state. But tumor suppressors, they act as recessive mutations. Now, what that means is you have to lose both of the homologs inside you. And the reason familial inherited breast cancers happen is that a loss of one of these two genes, the homologous genes, is something that takes a while. But once you've lost one, the chances of losing the other are now tremendously elevated. And this is probably because of DNA mismatch repair, the fact that you've got this other gene as a potential template. And so the loss of one, that can be a fairly slow process. It may take years before you would lose one. But then the loss of the second can happen within a much shorter period of time.
So it's like once you finally lose the one, then you can lose the second one very quickly.
Yeah.
Okay.
And so here is why these are part of familial inherited cancers, is that if you've got both functioning copies of the tumor suppressor, your chance of cancer is fairly low. You have to mutate one, and then you have to mutate the other, what's referred to as the two-hit hypothesis. If you start out life with only one, you only have to lose that one. And that's going to happen at a much higher frequency. So that's why there are screens for different tumor suppressors associated with different kinds of cancers. And if you have a mutation in one of those, then your chances of developing a cancer later in life are much higher. Probably if you have a mutation in both, you don't make it out of the fetal development stage. So nobody is missing both of them and able to get up and walk away. But this two-hit hypothesis makes it so we've got to have a number of events that happen. And when you look at it from a perspective, the number of events that have to happen is in the 5 to 10s to dozen different mutations. There's actually a now fairly old study on the progression of colon cancer. And the number of mutations you have to pick up is not insignificant. So in order to have a cell go into the full-blown transformed state, you have to have a specific oncogene mutation. That means it can be one of a handful of oncogenes, but the mutation itself has to be very precise. It's got to be an activating mutation. That's going to act as a dominant mutation. Additionally, you have to lose both of a specific different tumor suppressor genes. And so we've got a wide range of different kinds of cancer types because there are different kinds of oncogenes. It's one of the things that back in the Richard Nixon administration, they began with something called the War on Cancer. And it did fund a lot of different research, but the idea that cancer was one thing is, in fact, it's not quite naive. There are a whole range of different kinds of mutations that can occur in a whole range of different genes to generate a whole range of different kinds of cancers, and different ones require different kinds of treatments. Now, we've been talking a little bit about some generic chemotherapeutic treatments, things that are going to block the spindle fiber formation. Those are fairly generic, they work, and again, what you got here is cells that have become transformed. They are dividing much faster than your standard cells. So something that is going to be a metabolic toxin, something that will block mitosis, is going to be a kind of chemotherapeutic. I mean, a standard way to image potential tumor cells is feeding somebody a radioactively labeled glucose molecule. And of course, all your cells are taking up glucose, but the tumor cells are particularly active. They take up a good bit more of the label, and they will absolutely light up a PET scan. So, let's see. So you picked up mutations in an oncogene. This is going to cause the cell to begin dividing more rapidly. You may begin to have a slight loss of contact inhibition. You may start to see a little bit of piling up, but you might not have all the mutations necessary. You generate a benign tumor. Now, over time, the target for that cluster of cells that have picked up that mutation gets bigger and bigger and bigger because they are dividing faster. That provides the opportunity for another mutation to occur, and then still another mutation. And so, probably at a minimum, it's going to be 3 different hits, and probably more than that, to generate a full-blown tumor. This is why anything that is a potential mutagen is automatically a potential carcinogen. And pretty much every carcinogen is a potential mutagen. I think about this, like the 4-ball esters are probably not mutagens per se, but they are activating different kinases through receptors and things. So we have to be thoughtful about that. Now, we can tie this back in to the extracellular matrix discussion, in that as tumors continue to develop, they can undergo a phenomenon known as metastasis. This is what's known as a metastatic tumor. That's a pretty nasty thing. Metastasis occurs when cells of the primary tumor lose their adhesion molecules. They begin to actually lift off of the epithelial surface. And additionally, cells that are metastasizing can begin secreting enzymes that allow them to effectively burrow through the extracellular matrix. So you have an epithelial sheet, and one of these guys is transformed. They actually can physically begin to move through and invade, burrow into the extracellular matrix and make their way into a lymph node or a blood vessel where they can then go to another site. So this is one of the hallmarks of advanced stage cancer, is that you've got now not just a single tumor, but tumors now that have begun to spread throughout the body through metastasis. Yes, ma'am?
So if they've lost their adhesion capabilities, how are they able to stick onto a new place?
What they have done is they've lost their cell-cell communication ability. This is the question is, how can they begin to invade and then move on to the next step? They've lost their cell-cell connection ability. They now lift off and begin moving around, and they will lodge someplace else. And then they can, again, burrow in and kind of get into different cell types. And, on one hand, we make this sound like, it is an intentional thing. What's happening is that a tumor cell is mutating and by microevolution in a way, picking up mutations that allow it to divide faster. Another thing that very often will happen is that tumor cells will begin secreting a hormone that promotes vascularization. And there are a number of hormones that actually will cause the differentiation of blood vessels. And so a standard thing with a tumor is it can be phenomenally vascularized and actually difficult to remove surgically because it has so much blood flow. It turns out that there are hormones that also will block the development of blood vessels. And apparently once you reach, 21 or so, you really don't need a whole lot of new blood vessels, and you can actually take this as a therapeutic. Another really interesting therapeutic that has been around for a while are classes of kinase inhibitors. And Gleevec is one of these specific ones, in that you can inhibit the kinase in specific kinases, and with certain tumors, effectively make it so the tumor stops. To the point that you can begin treating cancer not as a immediate kind of problem, but as a chronic disease. You just take your tumors, your kinase inhibitor every day, and you're fine. Not a lot, that's one of the ideas. It doesn't work for a whole bunch of tumors, but for those that it does, it is absolutely a miracle cure. Now, as we're saying, burrowing through the extracellular matrix, you might ask, what the hell is a cell doing burrowing through the extracellular matrix? And I'll tell you, there are a whole class of genes that you typically don't have being activated once you have become a baby, or once you've been delivered. You're no longer a fetus. And specifically, there are classes of genes that are involved in embryo implantation. So, not to go into it too much in the weeds, but you have the oocyte released by the follicle, and the oocyte gets fertilized. The fertilized zygote, now after a few days after fertilization, makes its way down the fallopian tubes and into the uterus, where it then actually is going to embed itself into the uterine lining. And it does this by secreting a set of hormones that are responsible for effectively opening up and breaking apart the extracellular matrix, very tough epithelial lining of the uterus, and plants in there. And once it does, it then begins to vascularize, you begin to form the placenta, and you start to have a baby. So it's not like these cells are pulling magic tricks out. They are simply using the different kinds of functionalities that cells have. It's just that you don't typically have cells that are embedding themselves into the extracellular matrix other than the very early developing embryo. And the same thing with a variety of different vascularization cues, all sorts of things. So that is our cancer discussion in a nutshell. The critical point is an oncogene is acting as a dominant, a tumor suppressor is acting as a recessive. A dominant mutation means you only have to mutate one of the homologs. Got to mutate it just so, but just one of them. A recessive mutation means you have to mutate both, but that recessive mutation can be a whole range of things because all you have to do is either put in a stop codon, delete the whole thing, some kind of exon-intron splice mutation. It's fairly easy to mutate a gene so it just doesn't work at all. And that's pretty much what I got to say. So, as you make your way through your world, reading things, you're going to go, hey, I know what an intermediate filament is. Or, I know what is happening with a kinase inhibitor, why that might be useful. Or I understand a little bit about self-signaling to a point that I can go, if we have cells arranged in a cluster and one cell is secreting a specific hormone that can be picked up by the other cells, that's why those cluster of cells can differentiate. You know a lot of stuff now, and use it in good health. Good luck. That's it. Normally on finals day, we'd meet in here and I'd have donuts. That's been my tradition for a long time. You're going to be doing your final in your room or something. I don't know where you are. And so, good luck, and I'll catch up with you later. As I said, I'll probably drop another video on Sunday just to kind of go over a little bit of what to expect for the final, but, you know, it'll be open book, open note, blah, 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 blah. All right, dudes, that's it. I'm out. Bye-bye.